Right, okay, let's get started. So we're going to talk about genetic diversity today, eukaryotes, the difference in terms of how DNA is stored in eukaryotes and prokaryotes. Anybody tell me a difference, please? Yeah. Yeah, so longer, whereas prokaryotes are shorter in terms of the DNA. Yeah. Yeah, so obviously longer and they're wound into chromosomes, whereas in prokaryotes, they're stored as what, please? They're coiled. They are coiled, but isn't wound around histones, so not wound around histones. Anything else? That doesn't explain about the differences between the DNA. Well, basically DNA is linear in eukaryotes, whereas DNA is circular. What about also mitochondria and chloroplasts contain DNA? Have their own DNA. Anything about bases? No. Oh no, actually I'm thinking of mRNA there, aren't I? Okay. Right, so how would you define a gene? So what is a gene, please? What's a gene? Yeah. Yeah, sequence of DNA process. Good. Sequence of DNA bases codes for a polypeptide. Peptide. Okay, good. Okay, sequence of bases, polypeptide. What determines the primary structure of the protein? Let's talk about proteins. I mean, this is very linked to biomolecules. So this is really linked to biomolecules. They could ask you lots of things about structures of primary, secondary, and tertiary structures. So primary sequence of amino acids. So proteins, primary structure is a sequence of amino acids. Determines our folds. It's the order of bases that determines the amino acids. For a polypeptide, determines sequence of AAs. Somebody tell me about the genetic code. So in terms of the order of bases, so it's a triplet code. How would you talk about that triplet code? Codes for amino acids. What's it known as? Yeah, degenerate. Well done. Very good. It's a degenerate code. Oh yes, degenerate meaning there's more than one amino acids. So more greater than one AA. Oh no, greater than one triplet code. Triplet code. No, is it AA? So degenerate means that there is, yeah, there's more than, there's greater than one triplet code. So for to code for AAs, for an AA. It's universal, well done. In other words, it's found in all organisms on Earth except for viruses. Universal. Okay. Ooh, let's talk about actually DNA. So coding for proteins. So when it codes for proteins, in eukaryotes and prokaryotes, what's the difference in the code between eukaryotes and prokaryotes? Yeah, so they have introns and exons, whereas prokaryotes have what? Introns. So very good. Okay. They have exons, you're right. So they only have exons. When are introns removed? So those are removed in just after translation, transcription. My God, I'm all over the place today. This might not go on YouTube, this one. So removed just after transcription. Let's talk about protein. Protein synthesis comes up all the time. Okay, and I think we could just do a very quick summary of protein synthesis. Okay, transcription. Right, so the first stages, okay, a transcription is you're making an mRNA copy. So mRNA copy is made. What's the first thing that happens, please? What enzyme is involved? No, before that. RNA polymerase. So RNA polymerase attaches to the DNA.
So they have to separate the sides of it.
Wait a second. You do? Yeah. Oh yeah, I suppose, yeah, you're right. Oh, for God's sakes. Thank you. Okay, DNA helicase, first of all. DNA helicase separates the strands. So breaks hydrogen bonds. It separates strands and then uncoils. Right. So then RNA polymerase lines up the free nucleotides. So free nucleotides joined to the template strands. To template strand. like that. Okay. Okay, and then the mRNA strand is another step, but I'm just going to briefly go through this. mRNA strand is produced. It kind of peels away, and the DNA reforms to reform the double helix. How does the RNA polymerase know when to stop? How does it know? How does any enzyme know? Yeah, it will reach a stop codon. So stop codon, that then means that the RNA strand peels off a single-stranded piece of mRNA. Okay. Okay, there we go. Okay, before it moves out of the nucleus, so before it gets out through the nucleus, what's going to happen to it? Or mRNA. It's got to be spliced, yeah, spliced. Okay, so it's spliced, so it removes introns, non-coding parts, repeated bases, the VNTRs, all that malarkey. Anyway, and then it can travel through a nuclear form, and it then comes to ribosome. Here's the ribosome. So we're going to talk about translation now. So translation. Okay, and this clover leaf thing with an AA on it. What's that called? What brings the amino acid? tRNA. Okay, good. So tRNA, transport RNA, it has attached a specific amino acid attached. It's got a specific name for this part here. What's that part called? Well done. It's an anticodon, and the 3 triplet codes on here, that, they're known as codons. So it's complementary anticodon to codon, and then it attaches, it's complementary to the codon, and then the next tRNA comes, and so forth and so on. How the amino acids join together? What joins them together? Biological molecules. 1, peptide bonds. Thank you. Peptide bonds. Very good. There we go. Very simple. Expect questions where they'll give you codes. So they might give you a code, okay? And then they want you to do, that might be DNA, that might be mRNA, that might be tRNA, okay? And they want you to match up. They want you to match up the corresponding complementary codes. So let's do these together then. So what's the complementary codes for mRNA, please? What gets replaced? T to U, isn't it? So anyway, what would that be? Good. tRNA? Well done. There you go. Very good. They would expect you to know that. Very impressive. Oh, genetic code is not just degenerate and universal. There is another thing. It's non-overlapping. I forgot to say that. Code is non-overlapping. I don't want my YouTube subs having a go at me for missing something out. What are they likely to do? This is Dr. B's top tip. So Dr. B's top tip, they're likely to give you lots of data. They're likely to give you interpret base sequences. They love you interpreting base sequences. They give you the amino acids. Do you need to know the amino acids? No, don't revise amino acids. Don't revise the names of the amino acids. It's worth knowing things like the stop codon, but they tell you what the stop codon is. Anyway, there's lots of stop codons. AUG is one, for example. Okay. Good. Right. So that's genetic code. So much of this. We're not going to have time, I'm afraid. Oh, I do want to do classification. Can I do? No. Okay. All right. Well, no. What test? No, because it's paper 1. Okay. So, yeah, I'm going to stop there. I think there's some classification, investigating genetic natural selection and stuff like that as well. Anyway, that was half of genetic diversity. I hope you enjoyed it. I'll see you soon. Thank you. All right. That's okay. All right.